Greetings, professors, doctors, and fellow students. I'm Oscar, and this is the story of macrophage in liver cancer. To begin with, I would like to welcome you all to the field of cancer immunology. And this field is all about one observation. Prolonged inflammation often have a higher risk for cancer to develop. For example, bronchitis to lung cancer, hepatitis to liver cancer. So having a sustained state of inflammation is definitely not a good news. As there is a lot of types of inflammatory cells, which one is the bad one? So we set up a set of criteria to try to identify this bad cell in liver cancer. And this cell has to be expressed dominantly in the tumor environment, and they has to be able to secrete substances that can stimulate the tumor growth and the movement. So we come up with this candidate called the macrophages. And here is our experiments. So as always, we begin with human. So we collect about hundreds of liver tumors, and we perform a specific staining to visualize these macrophages. So similar to this model, we calculate the numbers of these macrophages in each tumor, and based on these numbers, we divide our 100 patients into two groups, the high and the low cell count group. And interestingly, we find the patient with a high numbers of macrophages in the tumor have a significantly shorter lifespan than those patients with a high numbers, with the patients with a low number of macrophages. So this tells us the macrophages is bad for liver cancer. Since human is a complex organism, we further move on to a much more simple model, the mice. So the color patch here represents the signals from the liver cancer. And what we did here is by injecting the mice with the macrophages, and 5 weeks later, we can see the mice here, their tumor size are much more larger than those that do not receive the injection. Moreover, these liver cancer also are more aggressive, that they spread to lung to cause the lung cancer. So now we know that the macrophage is bad because they can increase the tumor size as well as the aggressiveness. But how do they do it? So we identified one of the products that are produced by these macrophages called the CCL22. And this CCL22 can increase the movement of the cancer cell, making they move more vigorously. And more importantly, they decrease the level of the proteins that hold the cancer cells together. So in other words, the cancer cells are more easy to escape from the environment and to spread out. So in summary, I have told you that the macrophages is bad because they decrease the lifespan by increasing the tumor size as well as the aggressiveness through this product. So if we can stop these cells or perhaps these substances, we can demolish the liver cancer. Thank you.